Hi, good day and welcome to another day with CSEC Biology with Mr. Wilson. Today we'll be testing a leaf for starch. I'll be taking you through the lab, explaining some key steps in the lab. So you want to watch this video in its entirety. So let's just hop on into what we're expected to cover before the lab, and then we are going to be looking at the actual lab, and then you are going to go on and do the write-up for the lab. So let's go. Now, things to remember. You must record the date the lab was done in your lab books. So always remember the date that you were given the lab and the date that the lab is done must be in your lab book. The other important point is that all the pages in your lab book must be numbered. Don't forget these. These are very, very important points. Now, make sure you know which skill is being assessed. And why you want to know which skill is being assessed, it is because for the ORR labs, they would be written up a little differently than you would have planning and design. And I want you to check out my ORR video in the description below for more information on how the lab should be written in your lab book. Please remember, there are some guides to writing up each of these skills. You'll want to check out those videos. The procedure for all labs except planning and designing is written in past tense. Hence, you see the reason to know which skill is being assessed. Labs except planning and designing, implementation and drawing usually take the following outline. So labs except planning and design, implementation, drawing, they usually take the following outline. And let's look at it. Now, to report the lab, you are going to first, you need to have your date, followed by your title, followed by the skill, aim, apparatus, material, your procedure, diagram, observation, discussion, and conclusion. Now, you might wonder why this is formatted like this, or the color is here. This colored writing here is just to remind you as to what we would refer to as apparatus versus material. However, the indentation here is a reminder that when we write our procedure, we want to step away about 1.5 inches or 2 inches away from the left margin and have an indentation there for the teacher's report or your supervisor's report if you were in the real work world. Now, the diagram usually is not so much indented as these words, so the diagram could find itself in the middle of the page. But for your procedure, your observation, discussion, and conclusion, they are going to be somewhat indented about 2 inches away from the left margin. And to ensure that it is really, really neat, you could just use a pencil and a ruler to make a measurement and draw a very dull line. When you're finished writing up right beside this line, you are going to erase the line. All right, let's move on. Now, the date was the first thing we made mention of. Now, this represents the date the lab was done. And we're assuming here that the date the lab was done is actually the date that the lab was given. All right, so we don't want to get a lab and be sitting on it for days, and then we get to a point where we don't even remember what we were asked to do or the date that the lab was given. So once you're given it and you're given your timeline, the deadline, that sort of a thing, make sure you follow the guidelines of your teacher. The title. Now, this represents a topic from your syllabus that the lab captures. So, for example, if you were testing for starch, then it would come directly from photosynthesis. So, that type of thing, you want to say, what are you looking at? What topic are you looking at in your text now? What is it that we're studying now, right? And then that would indicate where your lab the direct relationship with your lab. And your teacher could help you with that if you're not sure. Skill. Now this represents the lab being tested. What skill are we looking for testing under this particular topic? It could be ORR, MMPD, implementation, AI drawing, you name it, whichever one. But you must know from your teacher what skill is being assessed. Then your aim, what is it that you intend to find out, what you want to find out? And this usually begins with a to determine, to examine, right? Usually your aim is what you intend to find out. What is this research about at this level as your SBA CXC labs? Now the apparatus material is really interesting. Really interesting. Now there is a slight difference between apparatus versus material. Now apparatus are those things that are able to be reused in the lab, such as your measuring cylinder, gradiated cylinder as it is called, forceps, metal boiling tube, old, and that type of thing. The list continues, dropper, so on and so forth. These things will be used in another lab. We're going to be cleaning them up, get them all ready for another lab. But when it gets onto material, these things are what we say, they won't be able to be used again in the lab. Once we use them, we dispose of them. For example, if you look at reagent, for example, like iodine, once it is used, we are going to just throw it away. So what we put on the leaf, or what we use to stain that cell that we're looking at, the organelles, we are not going to be collecting that iodine again. And once it's contaminated, we use it. We really don't use it again. The alcohol, to some extent, once used, you know that we're going to immerse the leaf in the alcohol, and then it's going to be disposed of. The splint, the splint is like a matchstick. So once it burns, then we are going to be it's just burnt. It can't be used again unless you have a little piece that you can relight until it's all burnt out. But usually we don't. We usually have some water, and once we use water in the lab, we usually dispose of the water that was used. I don't know that water is really considered really under the material stuff as such. You can leave me a comment on that, but definitely it is not an apparatus, so it will be more suitable to be placed in the material group, as we are looking at material, as we are looking at chemicals and reagent used in the lab. Hence we put the chemicals and the reagent under the material. Now, important to note, I have at the top here, look at the screen, I'm using my cursor, numbers, because number refer to those things that we can count. So you want to say how many of these you are going to be using. And over here I have number or amount, and this is because we are not going to measure the number of water. It's the amount of water, because that comes in volume. So we want to have an appreciation of that. Let's move on. Now, your procedure or the method. Procedure or the method. This is what you did if it's an ORR lab. And remember, you need to hop on over to my laboratory skills, those video, and they will explain this part of your lab real nicely. All right. A diagram. Your diagram usually falls right after your procedure or in front of the observation once you are going to be explaining things in your observation. So diagram, it shows the setup of the lab and any key point in the lab that will be captured in your observation. And for those persons who have ever seen a lab or done a lab, and you're, for example, you have ever seen, for example, the diffusion lab with hydrochloric acid, that gaseous thing, and ammonia, you would have an appreciation for key point. Where did we find this ring in the glass tube, that type of thing. And we're going to get there, so you want to follow us with these labs. We're going to be showing you all these labs, and the labs are going to be real labs, so we're not going to edit it so much. We are going to try to see if we even can capture the time what a lab is like really. You watch it and you get this real feel I mean, even sometimes you might be hearing some correction and so on. The real feel of what a lab is like. I'm not going to make it be one of those edited virtual lab type of thing. We're sort of running away from that. We want you to have a real feel of the lab. So you want to follow us, all right? Observation. What did you observe? And remember, we spoke about in that observation video we did on lab with lab rules, we spoke about observation pretty much being linked to your 5 senses. So in this particular lab that we'll be looking at, we want to look at see and feel. Now from where you are, you won't be able to feel, but the presenter might tell you what the feel is like, but you'll be able to see. So you want to follow in this lab, look at what is happening. Make sure you take note of the colors, the color change, and probably change of state. All right, solid change into liquid, liquid change into gas, that type of thing you want to make sure you make note of, right? Probably the boiling scenario between the alcohol versus that of water, that type of thing you want to make sure that you have a good grasp of. You're seeing, you know what is happening, and you're not relying on second-hand information. It is right in front of you and you're making the best use of it. What happened to the leaf? There are 2 leaves. What really happened? You really want to ensure that you're looking at the lab and you're capturing accurate information for your observation. Now, discussion. I like to say to my students, your discussion is your which, where, why, when, how, and what. And these are explained. All right? Your which, where, why, when, how, and what. And then we explain all these in the discussion to pretty much bring some value, some understanding to what we actually observe. Why we had to turn off the flame when we were putting the boiling tube in the water bath. Why we had to put the boiling tube in the water bath. What happened when we placed the leaf in the alcohol? Why did this happen? Right? So you're going to be seeing all of that. Remember, your discussion is always your which, your where, your why, your when, your how, your what. And bear in mind, it is not all lab you will be able to capture all these 6 which, where, why, when, what, that type of thing. You won't be able to capture it in all labs. But for some labs, you might be able to capture all. So just make sure you are doing the relevant thing and you don't try to stress yourself to say, but I didn't get how, or I didn't get why, or I didn't get what. Make sure that there is actually a which, where, why, when, what. And you could have this discussion with your teacher to see, to figure out if there's something that you're missing in the lab. And they, no man, you have not looked at the what. Let's deal with the what. Your teacher will nicely help you with that. Remember, your teacher should always be a part of your guide to success. All right? Even though you're watching my videos, always go back to your teachers. Always go back to your syllabus. Always ensure that you get a textbook, that type of a thing, to ensure that you're well prepared, you're well rounded. All right? Good. Let's move on. Conclusion. Conclude. It means that we're pretty much at the end here, or close to the end. What it actually means for the lab, however, is your final position. And this final position is based on your aim, what you intended to find out. Right? Now, in some labs, it may be any of the following. It could be a word. It could be a sentence. Or thereof. It could actually be a number or numbers. And it could even be, of course, a combination of. Now, I have seen in many labs where students have written, I have reached the point, try to move away from personalizing the lab. If you wanted to find out the number of persons moving through arbitrary between 9 o'clock and 10 o'clock, and it is 50,000, the conclusion is 50,000 persons pass through arbitrary during that time. That's what you found out. All right. You don't need to write I have found out or whatever, that. Don't personalize the lab. All right. Try not to personalize the lab. Let's move on. Now we are now going to be looking at the actual lab. The actual lab. So you want to grab a seat. You want to ensure that you call up a friend, and you want to make sure that you are watching just about everything. You have your book, you have your pencil, you are taking notes, you know exactly what is happening when. All right? So sit back, grab your pen, pencils, and let's go. Today we'll be testing 2 leaves for starch. First, we're going to be looking at the variegated hibiscus leaf, followed by another leaf which is very, very popular in Jamaica. It's pretty much invasive. This leaf is called the caronilla leaf. They call it bees bush in Jamaica. They call it, some persons call it rice and peas, but it bears this pink flower. Now this leaf will also be used in our drawing lab. So pay attention to both leaves. One is variegated while the other is not. All right.
Due to COVID, I'm home. I'm actually doing the lab from the stove in the kitchen. I actually got a beaker to borrow from school. So we're doing the lab at home so that our students can have an appreciation. So if we were at school, this setup that is on the stove would have actually been this. It would have been the Bunsen burner under here, with the tripod, that 3-legged stand, wire gauze, and the beaker on top with water. But due to COVID, we are unable to do that, so we have done everything we can for our students. We took these things home, and we are still carrying out the labs so the kids can see it online and be able to have an appreciation. Okay, so this is this setup at home. We would like to know the apparatus, what has been used for the lab. So I'm going to be showing you here. We have a test tube rack with a boiling tube. The tube is much bigger than the test tubes, and we have some measuring cylinder around here. And right here we have we like to call it a tweezer. We use this to remove the leaf or spread the leaf on the tile, the white tile. It's not a tweezer, I think it's called a forceps. And then here we have iodine. We have a wooden splint, and the wooden splint is what we would use to light the Bunsen burner. So you would just simply ignite it here. I would come on over with a splint here. And if we remember anything, we remember that oxygen will relight a glowing splint. So that's a glowing splint. If it should get some oxygen, then it will just start blazing again. Alright, that is a lab by itself. And then here we have, now there's always some confusion, this being the test tube holder versus this being the test tube holder. In some cases, this is called a test tube rack, and this is called a test tube rack, and this is called a test tube holder. But this is actually used to hold the test tube when it's hot.
Metal test tube holder, while this is the test tube rack.
This here is the dropper. We're going to be using it for to apply the iodine to the leaves as soon as we are ready. So I'm using one hand to tape and one hand to show you the stuff. So I'm hoping that I'm thinking and I'm doing just about everything right. So I'm going to be putting in the variegated leaf now into the water. You can see that the water there is all boiling, and I'm putting the leaf in the water here. One other thing that we want to explain, the first thing we wanted to do, in a matter of fact, was to ensure that we have an appreciation of a color of the leaf. This is going to be our first observation, so we need to write this down. What is the color? And then at the start, we needed to know what was the color of the water, because we really don't want to make any mistake. So here, the first step here is to find out why we had placed this leaf in the water. So you have to figure that out or do some research. All right, we're going to continue. So that one is in the water, and you need to tell me why it's in the water there. And then we are going to do the same thing with the carinilla. It's in the water as well, boiling water. So you know what will happen if your finger get in there. So I am at home. I'm praying that I won't get myself in any trouble before the end of Corona, carrying on chemicals to do labs on my stove and to assist you. But I think God will see us through. All right, so we're going to remove this now. So you will remember when we removed the variegated leaf from the water, it had the clear color just the same. But having placed the carinilla leaf in the water, you observe that the color has somewhat changed. Now this is a part of your observation that you will need to record. Now we're going to move to the next step of the lab, and we need to turn off the flame. So look, the flame will go out. So with one hand holding the camera and the other, I'm pouring some alcohol. Into the boiling tube, and then I'm going to be putting the variegated leaf into the boiling tube. You want to ensure that the leaf goes right down into the alcohol, and then you want to put this now in what we call a water bath. So the beaker with the hot water there is called a water bath. And now we're going to be putting the boiling tube with the leaf. The leaf is in alcohol in the boiling tube, and we're going to place this now into the water bath. So it's in the water bath there, and we're going to allow it to stay there for a little while, after which we are going to remove same and make another observation. So just let it stay there for a little while. I'm going to come back in a few to show you what happens. Remember, when I placed it in the alcohol was pretty clear. It had a color of water, so to speak. Some would say water has no color. This is an important part of your observation. If you observe the fire is off, but if you look in the boiling tube, you see what's happening? What's happening? Can you tell me? It shows sign that it is boiling. Now, yes, it is boiling. There it is. And why is this so? Alcohol water boils at 100 degrees Celsius. However, alcohol would boil at 78 degrees Celsius. So if alcohol is boiling at a lower temperature, if you are supposed to expose the alcohol to open flame, tell me what you think would happen. All right, so that's a part of your discussion. Why wouldn't you put this alcohol over open flame? Then the next part you want to look at is, why is the alcohol boiling? That's a part of your observation, and for the discussion you are going to explain. You are going to do the why in the discussion. So for safety reason, I'm holding this away. I'm holding the camera with one hand. Now that's another bit of observation. Look at the color of the alcohol. Can you see? Look at the leaf. Now another thing that we want to do, we want to feel the leaf to see what it feels like. Now pretty much this is what we had, right? Pretty soft. There we go. Pretty soft. This was the original. This is where we started. Now we're going to be looking at the leaf. We're going to be looking at the leaf. Now the leaf is out. This is what the leaf looks like. Remember what it looked like earlier. And another thing, you wouldn't be able to determine this from where you are, but it is crispy. It is crispy because it would have lost some water to the alcohol. So now we're going to be applying iodine to the leaf, so as to test for the presence of starch. And if starch is present, we're expecting to see a blue-black color. Let's go. So we're applying the iodine to the leaf now. Now, wherever there is starch, remember it's a variegated leaf. So some parts of the leaf had a pretty light color, while some part had a green color. Now look at what is happening. We're going to give it a little bit of time, and then we're going to look at it, and you are going to do the write-up for this lab. Right? So we're testing a variegated leaf for starch. So let's give it a few seconds and then we'll get back. Alright, that's the color of the alcohol we're using. You have to watch this video because I'm going to be flashing the name, the correct name for some stuff at the top. Alright, and here we have the open flames here. We're going to turn it off. We're going to place the leaf in the boiling tube with the alcohol. Look out for the name that we're going to be using. And then we're going to be placing this boiling tube with the leaf into this water bath. Now it's a hot water bath. This is not a hot water that you would want to have a bath in. Alright, so we'll put it in there. We're going to be looking out for something. Remember when we looked at the variegated leaf, we said then that the alcohol, from our observation, the alcohol started boiling. Let us see what will happen with this leaf. Alright, there we have some motion. Something happening there. Something happening there. Alright, while we wait on that, just let us look at the experiment at hand. Now this is the variegated leaf. My lighting is not so good, but you will observe that there are you have parts of a leaf that is pretty light in color and parts that are really dark. Now the dark area there, while my camera doesn't give you the best of color, that is the blue-black color. Now you are supposed to tell me now that light color, would you say that there's starch in that area? Remember, wherever there is starch, you should see a blue-black color. Now, not everywhere on the leaf we're seeing a blue-black color. That dark color that appears to be black to you, that is a blue-black color. Alright? So, make the determination for me. Please do not do this experiment at home. We're having some boiling taking place there. In your chemistry class, you'll tell us what's boiling. So this is the finished product. The leaf is still in the alcohol, and this is the color there. Not the best at camera, but I'm thinking that you are seeing the color. We're going to be removing this now. Put it in the hot water. Just immerse it in a hot water bath, and I'll tell you why in a bit.
So the leaf was immersed in hot water, or it could be immersed in any water, to soften it, because having removed it from the alcohol, it was crispy. So here we have the cornula leaf after the iodine was added to it, and this is the variegated hibiscus leaf, and this is what it looks like at the end of the experiment. Thanks for watching, and if you found this helpful, please remember to like, share and subscribe, and check out our other publications.